So for the first four here, okay, we want the formulas. Dinitrogen trioxide is a molecular compound because it's got prefixes in it, okay? So we need a 2 on the N because the prefix is di, trioxide, that's 3. Our formula is N2O3, that's a 3. There we go. That still doesn't look like a 3. It's supposed to be a 3. Okay. Number 2. We have calcium, which is a metal, so this is an ionic compound. Okay, when we have ionic compounds, we have to drop and swap to get the formulas right. Calcium's a 2+, chlorine is a -1, so when we drop and swap, okay, we end up with CaCl2. Okay, these are just 1 mark each, guys. There's no half marks. They either have it completely the way I have it, or it's not right. Okay, number 3, copper 2 nitride. So we got Cu, copper. And what does the Roman numeral 2 tell us? That it has a charge of 2. All right, so use the 2+ charge when we drop and swap. And it's with nitrogen, which is -3. So we'll have 3 here, 2 there when we drop and swap. Cu3N2. Okay, and the last one, magnesium sulfate. Also an ionic compound. Magnesium is Mg and it's 2+. Sulfate is a polyatomic ion, and its charge is 2-. Therefore, they cancel out. And we're left with MgSO4. Questions on the first 4? Sydney? I'll let it go today, but in the future, if you don't need a number outside of them, they shouldn't have them. Okay. Any other questions on the first 4? Okay. For the last 4, we're writing names. What should none of these names have? Right. Okay. Our first one is a molecular compound because it is both non-metals. So that is going to be sulfur tribromide. If they have a hyphen in there, that's fine. Okay. Number 6, we've got lithium with oxygen. That's an ionic compound, so we will need no prefixes. That is simply going to be lithium oxide. Okay, number 7, we have iron with nitrogen, but iron has more than one possible charge. So in order to figure out which iron it is, we have to look at nitrogen. Nitrogen's always a -3. Since they're 1-to-1, the charge on iron has to be +3. All right, so this is going to be iron, Roman numeral 3, nitride. Okay, and lastly, for number 8, okay, we've got sodium with nitrate, ionic compound, requires no prefixes. It's just sodium nitrates.
Sorry for the interruption.
So what we're going to be going over today, guys, are lab report assembly and expectations, okay, things like that for how do you write up and submit and all of that, your lab reports for Science 10, okay? Now, obviously, you're going to be submitting them all through Google Classroom. I'll give you a template for each and every one, okay? Basically, you just need to fill in the important stuff. The skeleton will already be there. Okay, so in that way, it kind of makes it a little bit easier for you. But we're going to talk about what each part of the lab report should look like so that when you're putting yours together, you know, you're much closer to the final product right away, as opposed to the first lab is a learning experience and I do crappy on it and then I do better after that. Doesn't seem fair to me. Okay, so we're going to go over, here's the expectations, here's what you should be putting together. All right, everybody with me there? Okay, so the first thing your lab report will have, okay, is a problem. Okay? And in the template that you get, it'll just say problem, and then there'll be nothing beside it, and you'll have to fill it in. Okay? What you would put there is the question or the problem stated as a question in your own words. Sometimes on the lab handout, which you'll get electronically again, okay, I'll have the problem in there in the way that I've written it. You will have to restate it in your own words. And it is like Jeopardy, you do have to state it in the form of a question. Okay? So an example, and this is the example we'll use kind of all through this, okay, is what's the effect of light on plant growth? Okay, if that's what I'm trying to, you know, if that's the problem I'm investigating, that's how I would write it. What is the effect of light on plant growth? Obviously that's pretty simplistic, but it's an example, it's supposed to be. Question? That's up to you. I would say it's probably not a bad idea to have the example down. Just so you've got something to refer to. Okay. Another example could be if I was, someone who's working in technology development, I might be doing an experiment on bulletproofing glass. Okay, and my problem might be, what is the effect of glass thickness on bullet resistance? Okay, or something like that. And then I would do an experiment with varying thicknesses of glass and shoot at them, see what happens. Okay. The next thing you're going to have is your design. And in your design, in the lab template that you're going to get, it's going to say, identify your manipulated, responding, and controlled variables and explain them. Okay? So you can't just say, the manipulated variable is this. You have to say, the manipulated variable is this because. All right? Now, quick review of the variables. The manipulated variable is the part of the experiment that's being changed on purpose by the experimenter. Okay? Essentially, it's kind of what you're testing. Okay, so the example here would be the amount of light that's given to plants. Right? So the experiment that I might run for this light on plants experiment might have like 5 different plants, and I would give each of them different amounts of light, starting with no light at all, moving to maybe like 6 hours of light, 12 hours, 18 hours, 24 hours, okay, and then see what the effects are. Okay, so the manipulated variable there would be the amount of light the plant receives. Now, if I was writing this in my lab report, I wouldn't just write amount of light given to plants. I would say amount of light given to plants because each plant is going to receive a different amount in order to test the effect. Okay, that would be my explanation. This is the identification. Okay, so this is me identifying the manipulated variable, and then I would have an explanation after that saying this is why. Okay, so there's always identify and explain. Okay. So we got manipulated variable. What's the responding variable? The results. Yeah, it's the thing you're watching. All right. My problem here is what's the effect of light on plant growth? So what's my responding variable? How much the plant grows. Okay, that's the thing I'm watching or the thing I'm measuring. Okay, here's what I manipulated, light. Here's what I'm watching for, plant growth. All right, so the responding variable is whatever changes as a result of the manipulations made by the experimenter. Okay, so yes, in other words, the results. If I was explaining this variable, okay, so I would identify it as the growth of the plants as the responding variable because plants receiving more sunlight should grow more. Okay, or maybe I would say the growth of the plants is the responding variable because this should change as a result of how much light they receive. Something in there explaining why it's going to, or what it's going to respond to. Why it's going to respond or what it's going to respond to. Everybody follow there? Okay, okay. What are controlled variables? Because that's the last kind. Right, okay? They are the things I have to keep the same or control throughout an experiment. If I don't, they could interfere with my results. Essentially, they're other things that could affect what I'm testing. All right? So in a case like this where I'm testing the effect of light on plant growth, other things that could affect plant growth need to be kept the same so that they don't have any effect. All right? So that would be things like I would need to make sure all the plants are the same kind. Okay, like I would do this on all tomato plants or all carrots or something like that. Okay, I wouldn't compare an apple tree, a spruce tree, a bean plant, and because they're all going to grow differently. They all grow at different rates. It's not a fair experiment. It's not controlled. Okay, other thing I would want to control: how much water do they get? It would hardly be fair to give the plant that's in the light, the most water, because then I'm not testing the effect of light anymore. I'm testing two things, and I'll never know which one caused it to grow better. Soil, same amount of it, same kind, okay? So I'd go out and buy like 3 bags of identical potting soil, okay? And make sure that they all got exactly the same amount of it so that soil wasn't a factor. It was the same for all of them, okay? They should probably all be tested in the same temperature. All of these things are things that could interfere with my experiment if I didn't keep them the same for every plant, all right? So controlled variables are other factors that could affect the outcome of the experiment if they were left unchecked. Okay, so in other words, the same plants, the same amount of water and soil, same temperature. Okay, and you would identify on your lab report 3 of these. Okay, you always list 3 controlled variables and you explain each one individually. Okay, we don't say same plant, same amount of water and soil, same temperature because they could all affect the plant. Yes, they could. Okay, same plants because all plants grow at different rates. Same amount of water because water can influence the rate at which plants grow and carry out photosynthesis. Same soil. Okay, soil is where plants get nutrients from. Soil needs to be the same for all plants. Okay, things like that. You got to explain each one individually. All right, now back to variables here. We've said you're going to list in any lab report 3 controlled variables. Is it possible you'd have more? Yes. Okay, in terms of your manipulated variable, how many of those can you have? 1. You can't test more than one thing at a time or you'll never know which one caused it or if it was a combination of the 2 things. Okay, so you can only ever test one thing at a time, but I see it happen on lab reports all the time. People list 2 or more manipulated variables. There's just no way. Or they don't list 2, but then they design an experiment that has 2, okay, without realizing that that's what they've done. Yes, absolutely. You could have more than one responding variable. That is entirely possible. Okay, if we're doing an experiment where we're calculating the acceleration of a cart going down a hill, there's going to be several responding variables like distance and velocity are both going to change as it goes down. So yeah, it's entirely possible to have more than one. We try to have only one, but sometimes there's more than one. Okay, questions on the variables? Okay. Now, the next thing is your hypothesis. Okay? A hypothesis is probably the most important part of an experiment. Okay? It's the thing that tells us what you think the relationship is in the problem. Okay? I don't like to use the term answer to the problem because it's really not. Okay? A hypothesis is a deductive statement. Okay. It says, like for the example that we're doing here, okay, I would be talking about what's the effect of light. This is the effect I think light has on plant growth. This is what I'm going to test, and these are the results I'm going to get. Okay, that's what a hypothesis needs to tell me. It needs to tell me 3 things. Okay, here's what's going on, or here's what I think is going on. Here's what I'm going to do to test it. If I'm right, these are the results I'm going to get. Okay, so it's a prediction, not so much an answer. Okay? As a result, a hypothesis always needs to have these 3 words in it. If, and then. If, and then. Okay, so it's an if and then statement that communicates what the relationship, okay, between the manipulated and responding variable is thought to be. Right, so if we're talking about this particular experiment with the light and the plants, I would say something like this. If plants require light to carry out photosynthesis, and 5 plants are given varying amounts of light, then the plants receiving the most light will grow the most, or will show the most growth. Okay, so I've said what I think is true about light and plants. I've said what I'm going to do briefly to test it. I'm not writing out my whole experiment and the hypothesis. Okay? And then I'm saying, here's the results. Okay? That way, I can be right or wrong definitively as a result of my experiment. The worst kind of hypothesis is the politician's hypothesis. If I don't say anything or I say everything, I don't insult anybody. Okay? A hypothesis has to be, this is where I stand. And if I'm wrong, I'll come back and do something else. Okay? You can't try and please everybody with a hypothesis. It has to stand for something. It has to say something so that at the end of an experiment, you're right or wrong. No, no. In fact, if you do that, it's bad science. You're not going to lose marks if your hypothesis isn't correct. You'll lose marks if it's not in the right format or it doesn't talk about the right things. But if you put in a hypothesis and after your experiment it's wrong, then you say in your conclusion, these are the results I got. They don't support the hypothesis. Therefore, the hypothesis is rejected. I'll study this the next time, or further study in this area will be forthcoming, something like that. Yeah. You never want to go back and change what you said. It's better. Most hypotheses are wrong. In the real world, when you're doing actual science, like 90% of hypotheses are wrong. That's how you learn things, is by being wrong once in a while. Okay, so in the if part, so the if part here is kind of this part here. Okay, so what the relationship between the manipulated and responding variable is thought to be is the if part. Okay, so that goes with if. Briefly describing the experiment is the and part. Right? In my hypothesis there, I said 5 plants will be given differing amounts of light. Okay, that's a brief description of what I'm going to do. Right, so that's the and part. And then the last part, the then part, outlines your expected results. Okay, so like I said, hypothesis is the most important part of the experiment, in my mind anyway. Okay, your lab reports are out of 30 marks. 5 of them are for the hypothesis. Okay, it's worth 1/6 of the lab report. It's that important. Okay, your design, which includes your problem and your variables, is worth 5 marks as well. Okay. All right. So the other explanation here, the other part of this explanation is the example. Okay, so the example here would be if plants require light for growth and 5 identical plants are given different amounts of light, then the plants that receive the most light will show the most growth. That's a very simplistic hypothesis, okay, but it's a good example. It's absolutely crucial you get the hypothesis right, okay? The hypothesis is the part of the experiment or the part of the lab report that people mess up all the time, okay? Like they'll just put in for the hypothesis, even though we've talked about how it's an if, and then statement, they'll write in there, I think this is going to happen, okay? That's not a hypothesis, okay? That's a guess, okay? And we're not trying to guess here, okay? We want to say, here's what I think is going on, this is what I'm going to do, and here are my expected results because of that, okay? Also, we try not to use I, we, us, okay, terms like that in our hypothesis. We try and keep it objective, okay? I never use any reference to myself, okay? Because in the truest sense of it, if I become part of this hypothesis, if anyone wants to do my research, I have to be there, okay? This way, someone else can try and replicate my work without me. Okay? But if I put in there, and I use 5 identical plants, and I give them different amounts of light, then I've got to be there every time someone does this experiment, and I don't want to be. Okay? So we try to keep that part out of our hypotheses in general. Okay, so just again here to reiterate, okay, what's in the if part? The if part is what is the cause of the problem or the relationship being investigated, and briefly describes the experiment. Then what do you expect if you are correct? All right. Now, what usually comes after the hypothesis is the materials. Okay, but for basically every lab, I'll have already outlined those and it'll already be on the template. Okay, so materials is very rarely something you're going to have to write out or worry about doing. Okay, I think even on this one I already have, where you're designing basically everything on this one, I think I already have all the materials on the sheet for you, okay, or on the template for you. So yes, materials would usually be here, but it's not something you're getting marks for because it's not something you're ever going to do. Okay, next thing we should have, okay, is our procedure. for most of the labs we do, you will also not write this out. The exception, of course, being the one we're about to do. Okay? In this one, you are sort of designing the procedure. But your procedure needs to be explicit. Okay? What does explicit mean? Means leaves no doubt. Okay? That's why sometimes, like, you know, if you're buying a song off iTunes, it has the little E, the red E beside it, has explicit lyrics. It means it's not sugarcoating anything. It's not saying it cryptically. It's just outright saying it. Usually it means it's using foul language. But okay, in this case, explicit means it leaves absolutely no doubt. So for a procedure, what we mean by explicit is write it so that an incredibly stupid person could follow your instructions correctly and do the lab right. Okay? Assume that, like, a grade 2 kid is trying to do your lab. They need to be able to follow the instructions. Okay? If you don't outline every single little thing, assume that they will not do it. Okay? So they are step by step, not a paragraph. Okay? It is a numbered list. Step 1, step 2, step 3. If you write a paragraph, that's really hard to follow. Okay, as a set of instructions, a paragraph is really hard to follow because you're following, okay, all right, I'm going to do this one. For the first sentence, it's fine because it's the start of the paragraph. But by the time you're on like the 4th or 5th thing, you're halfway through the paragraph and you have to read the whole paragraph again to find out where you were. Okay, it's way better to have a numbered list. People can remember, I'm on step 5. Okay, go back to step 5. Okay, and we can do that. So it's always a step by step, okay, set of instructions. Okay? And those instructions talk about preparing and performing the experiment. Okay? And by preparing and performing, I mean essentially making sure that one of the biggest things in there is making sure your controlled variables are controlled. You have to tell people how it is that you're controlling those things. Like, for example, in the lab we're doing here coming up, we want to make sure we use about the same amount of all of the unknown materials. Okay? Having different amounts could affect how we interpret the solubility results. Okay? If I put like 2 grains of one thing in a depression and a big pile of one in another, that's hardly a fair comparison when I add the same amount of water. Okay? You guys follow me there? Right? So we want to try and in this experiment make sure that we have the same amount without actually like doing the mass, because that takes way too long. Okay? But about the same amount of everything. So part of that is making sure the controlled variables are controlled. Yeah. Okay, but like I say, for most of the experiments that you guys do, I'll already have 1, 2, 3, 4, 5 cookbook set of instructions that you can follow. Okay? Other things that need to be in your procedure, okay, it says preparing and performing, but you should also tell people what kind of observations they should be making. Okay? Okay? What kind of things should you be measuring, recording, making note of in some way? Because all experiments are different, right? If you're doing a physics experiment, you would probably be actively measuring certain things like distance or velocity or stuff like that. Okay? In a chemistry experiment, you might be making more like anecdotal kind of notes, like when these two things were added together, the solution turned yellow. Okay? That's more of a qualitative observation as opposed to a quantitative one that includes a number. Okay? You're telling people what happened. All of those are equally valid, okay? But you need to tell people, when you do this step, watch for things like this and write it down, okay? Or take this measurement after this step or during this step or whatever it happens to be, okay? Because if you don't say that, people will just run the experiment and go, that was neat. What did I learn? Nothing, because I didn't write anything down, okay? And then they won't have any data. Well, you can't perform the next step if you don't have your data, okay? All right, observations. That's your data, okay? You have to make observations. Okay, charts, graphs, okay, other data including anecdotal notes acquired during the experiment. Okay? Going in, you have to know what you're looking for. That's your responding variable. Most of your measurements and observations will have to do with the responding variable. Once you have observations, what do you generally do with them? Even before you make your conclusion, you have to do something with your observations before you can make a conclusion. You have to analyze them. You have to figure out, what do these observations say? Okay, that could be, it could also be that you make some graphs that show a relationship between some other things that went on in your experiment. You do some calculations, okay, things like that. Those things go in your analysis. So calculations, review, comparisons of data, okay. In terms of your lab reports, you'll be answering the questions or doing the calculations that I tell you to do, or making the connections that I ask you to make, okay. And you need to have detailed explanatory answers when you do that. Okay? So basically, you're figuring out what your observations say during the analysis part of any experiment. Once you've analyzed your data, and you now know what your observations and data are telling you, then you can look at, how does that relate to my hypothesis? So if I'm looking at the experiment that I was doing here with the plants and the different amounts of light, I had one plant that was in total darkness. Okay, that's kind of my control group. Okay, that plant in total darkness is my control group, because it's not going to do what? It's not going to do anything. Okay, I need to know what happens to a plant if it gets no light as a base kind of for comparison for all the others. Okay, I'm going to have a plant that gets 6 hours of light, a plant that gets 12 hours, 18 hours, and 24 hours. All right, that was my experiment. Here's the results I got. Plant with no light died. Plant with 6 hours of light grew very little. Plant with 12 hours of light grew a lot. Plant with 18 hours grew slightly less than the 12-hour plant. Plant getting 24 hours of light died. Okay, so I got to look at that, and what that tells me is that more light doesn't equal more growth. That's what my data tells me, because the plant that got the most light died, and the 18-hour plant didn't do as well as the 12-hour plant. That's me analyzing my data. Okay? Now, what does that say about my hypothesis? My hypothesis said the plants receiving the most light should show the most growth. Does it agree? Does the data agree? No. Okay, so I've done my analysis. My analysis says that plants that get more light don't do better than plants that get less light. Okay, my data seems to say that 12 hours of light is the ideal amount of light for plant survival. Okay, less than that is not ideal. More than that is not ideal. 24 hours, plant cooks. Okay, so now I got to come up with a conclusion. Okay, first thing I do in my conclusion is I restate my hypothesis. So that's easy. Copy and paste it. Okay, just go back up the hypothesis, copy, paste. Okay, bring it back down to here. Then I use my data and my analysis to explain whether or not I can accept it or reject it. Okay, so a good conclusion is kind of like a short paragraph almost, because you have your restated hypothesis, which I don't have here. Okay, and then you can see in my example here that I summarize my findings. Okay, the analysis is where you're going to find all the data and all the analysis, but in the conclusion I want to just have a brief summary. Okay, and that is, as it was found that plants receiving small amounts of light and very large amounts of light showed little growth, it can be concluded that plants require certain amounts of light and darkness to be healthy. Therefore, the hypothesis is rejected. That's still a fairly simplistic conclusion, right? But it does what you need to do, which is talk about your results and how they relate to your hypothesis, and then say, I can accept it or reject it. Okay? But this goes back to what we were saying about the hypothesis. You can't be a politician in your hypothesis. You can't sit on the fence. You have to put yourself out there and be right or wrong. Okay? This is what I think, and this is what's going to happen. If you're wrong, then you say it. Okay? And I've said it here in my conclusion. My hypothesis is rejected. It wasn't right. Okay? The worst thing you can do in a conclusion, because your conclusion is worth 4 marks, the worst thing you can do in your conclusion is this: I was right. Well, goody for you. Okay? It ain't going to get you any marks, okay, writing I was right. What were you right about? Was it today? Yesterday? When were you right? Okay? Saying I was right. What if you weren't right? Okay? You have to restate the hypothesis. Here's what I said. Here's what was found and how it related to what I said, and I was right or I was wrong. Okay? But we never write I was right or I was wrong. Okay? We say the hypothesis is accepted or the hypothesis is rejected, or sometimes you can conditionally accept the hypothesis. Okay? You could say, the hypothesis is conditionally accepted because there appeared to be parts of the hypothesis that held true, and then there was a point where the hypothesis fell off. Okay? This wasn't accounted for in the initial findings, or something like that. You can conditionally accept sometimes, but most of the time, it's either rejected or accepted. Okay? Everybody follow there. All right. The last part of your lab report is your evaluation. Okay, and that's not me marking you. You evaluate the experiment and you find in your lab report 2 sources of error. Okay, 2 sources of error and suggest improvements. Now, where do people go wrong when they're identifying sources of error? Where they go wrong is they identify human error. Human error can never be a source of error. Okay? The sources of error you identify have to be flaws within the experiment itself. Okay? So, for example, my physics kids were doing a lab yesterday where they had motorized carts running along the floor. Okay? A source of error for that lab is that the floor is not perfectly level or smooth in all places. Okay? So, they were trying to have a cart that went in a straight line at a constant velocity, but there's little bumps and humps in the floor that cause the cart to turn and speed up and slow down, so it can't do that. Okay? Was there anything they could do about that? Nothing. I told them, here's the floor. Run the experiment on the floor. They had no choice. Okay? A source of error? Floor is not a perfectly smooth surface. It's as close as possible, but it's not perfectly smooth. Therefore, it could affect the velocity of the cart. Okay, for an experiment like with the plants here, okay, a source of error could be something like there were several days during the experiment where it was overcast and direct sunlight was not available. This could have affected the growth of some of the plants because they did not receive direct sunlight. An improvement for that could be run this experiment under artificial lighting. Okay? Then you have consistent lighting all throughout. You don't have to rely on sunshine. Okay? That could be a source of error. There wasn't anything you could do about it. You can't control the weather, but it was something that could affect the experiment. Okay? What people unfortunately do is they write something like this. I screwed up step 5. You did? And you didn't go back and fix it? You just screwed it up and kept going? Like, what the hell? Like, who runs an experiment like that? Okay, who says in their sources of error, yeah, I messed up, and I was too lazy to fix it, is what that says. Okay, and guess what I'm going to do when I see that? I'm going to go right back to all this other stuff and go, well, that's out the window, that's out the window, because I know you screwed this up and you screwed that up. There's no way you got those results if you screwed up step 5. So you copied somebody else's stuff, and now your whole report is in question. Okay? You never want to say, I messed up. Okay? Or, yeah, we had trouble measuring this. Well, then why didn't you say something? Why didn't you get a better ruler or a tape measure or something? Okay? But never admit that you screwed up. Don't hide it either. Okay? If you screwed up, fix it. You can never run an experiment where human error became a source of error. Okay? If you know you messed it up, fix it. Okay? You can always come in and do a step in the lab over again. Okay? Come in, schedule help for lunch or something like that. Okay? But never write in the evaluation that, yeah, I didn't do that, or I did that wrong, because then you didn't do the experiment right at all. Everybody follow me there? Okay? Always find something that was a source of error that you had no control over. Okay, at least according to the way the procedure was written, okay, and then find a way to fix it, right? For the experiment with the carts on the floor, okay, what we could have done was maybe built a perfectly flat surface, maybe got some, nice flat plywood or something like that, and built a surface that we knew was perfectly flat, okay? That would have changed the experiment. All right, everybody see what the expectations are now? Okay, realistically, guys, putting a lab report together, once you get good at it, okay, shouldn't be more than a half an hour's worth of work. Okay, if it takes you more than that, you're probably doing just a really, really good job and you're being really meticulous about everything. Okay, but if it takes you more than an hour to assemble a lab report, you're taking way too much time. Okay, because realistically, you're writing your problem, your variables, your hypothesis, the procedure you're not going to have to write except for this lab, and you're getting class time for that. Answer some questions, or write down some observations, which you would do in class. Okay, do analysis, do a conclusion. Like, it shouldn't take that long. Okay, I've had people who come to me like, Mr. Couture, I worked on this for 6 hours. Really? What were you doing? Like you actually sat in front of a screen for 6 hours? Like, you didn't take breaks or anything? No, I worked on it for 6 hours. Then you're obsessing. Okay, you're obsessing over it. If you're spending that much time on it, that's not healthy. Okay, a lab report is not meant to be that much work. Okay, so do it and then bring it in and I'll look it over. Okay, if I think it needs some more work, then I'll tell you that. Okay, if I think it's fine, I'll say don't change a thing. Okay? Most of the time I have at least something to say, but okay, once you guys get good at them and you start bringing them into me in November and December when we've been doing lab reports for a while, lots of times I'll just let them go. That's exactly, yeah, don't change a thing, just submit it now. Okay, everybody follow me there? All right.